Those quizzes this weekend, I'm sorry, I just checked some things this morning and they weren't open. So I will take care of that. And I didn't get your points tallied in from Friday. It was a bad weekend. And I didn't add any points in for the extra credit yet. So I'm going to have a busy afternoon doing all the things that people are anxiously looking to see what their point total is. I just apologize for that. So if you see any of the other students, I'll send out an email, but you know, the quizzes were supposed to be open and they were not, so I'll take care of that as soon as I can, right after class today. There might be some little check mark I didn't do when I thought I'd opened them. Okay. Wow, that's loud. So there hasn't been any changes, really. Wow. I'm not sure this thing seems so loud this morning. Maybe it's my sinus headache with this big, you know, wow. I'm sure all of you have been sneezing all weekend, too. Yeah, so there hasn't been any changes here for iClickers. I didn't have an opportunity to add in the points that I normally would have, so I will take care of that this afternoon. Oops, that's right. You guys have the ones with the weird. Oh yeah, and I didn't get a chance to switch notes with you with another section. So let me go to that other section. It's like everything's on a run right now of bad. So I understand your notes somehow got horribly misformatted, so I'll go to another section. And I'll see if I can swap their notes in with yours so you're not struggling with that. Okay. Yeah. Okay. So on Friday, we finished up our discussion of phencyclidines and ketamine and phencyclidine. I mentioned salvia divinorum. I would say in these notes here, anything that's in red, it doesn't really, it's from another section's notes that I had put some red, highlighted some things in red to remind me of something, and I forgot to change it back. So don't be concerned if you see something red highlighted in your notes. So we talked about salvia divinorum. The big thing for you to remember is it's actually a member of the sage family. It's been used in by some tribes to medicine men use its hallucinogenic effects. We just identified the active ingredient in salvia divinorum, and it's just been named Salvinorium A. So people are still working on what exactly this drug is stimulating to produce these, when people smoke it, to produce these rapid, short-lived hallucinations. It doesn't have a shape, it doesn't have an indole ring like serotonin, doesn't have a phenylethylamine side chain like the phenylethylamines. So people are not sure right now. Some people are studying that. So the last group that are in Chapter 6 that we've highlighted are the cannabinoids. And I guess eventually I'll change this to cannabinoids. That seems to be what most people are using right now as a term to describe the psychoactive drugs that are in the marijuana plant. So if you look at the history of this plant, it has a long history. It grows just about everywhere in the world. It's a pretty hardy plant. And it has a couple of names. Marijuana was actually something that was started in the 1930s in the United States to make this plant sound more maybe dangerous or foreign to people. But for most of its life, or history, human history of it, it could be referred to as the cannabis plant. Or if you're growing the plant to get the fiber out of it, there's a certain way you can do that. Some people would call it the hemp plant. So those are all the same thing. It just depends on what you're growing the plant for. So if you're growing it using mainly focusing on the fibers, people have made you can make all kinds of products from the hemp fiber. In addition to people for centuries have either smoked it or eaten the plant material to alter their mental states. So I would cross out number 2 if you have your notes in front of you. There is the idea that there's been an overall increase in potency of the marijuana that people have smoked, but that's not always the case. If some people were actually smoking products in the 1960s from Hawaii or Thailand or places like that, they wouldn't have been using marijuana that was of lesser potency. So that's not a very accurate statement there. The same thing with item number 3. The problem with it is it's very dated. I think this is looking at going back to 2005. They're talking about how many people had said they'd used it in the past month. So we know that that's, in the United States, that's a pretty large number. If you look at the 2 main psychoactive drugs that people admit to using in the United States, alcohol is number 1 and marijuana is number 2. And then way below that, you get into some of these other drugs that we've been talking about, where you start talking about cocaine, amphetamines, illegal use of painkillers. You see a big gap in the number of people who are using it. So the top 2 are alcohol and marijuana. And globally, there's a lot of people that use the cannabis plant. In India, there are some religious sects that use it during their ceremonies. Indonesia, parts of Europe, it's very popular to be used. So in parts of Africa, people have been using it for a long time. So it's pretty much a globally recognized psychoactive drug. So if you look at what 3 kind of major categories, people have recognized it as a use as medicine. So this was kind of pointing out that obviously when marijuana was put on Schedule 1 in the United States, it wasn't based on historical evidence. For centuries, people have known that marijuana helps with certain kinds of muscle cramps, and so it was used sometimes as a tea to help with those pains from muscle pain. It's been used as a fiber source to make fabric and rope, and as an intoxicant. So all of these 3 uses were well known, and actually in the United States, George Washington had a hemp colony. By what I mean a hemp plantation. So if you want to grow the marijuana plant for fibers, you actually want to go with the male plants at that point. I'll explain that you can separate out male and female cannabis plants. The female plant is what you want if you want to get an intoxicant. I'm going to talk about where the resin is and the psychoactive ingredients are in the flowering bud of the female. If you just want to use the plant for fibers, you can just separate out and keep the male plants and let them get very tall. And if you crack that inner stem, you'll see that it's filled with wonderful fibers. So George Washington's plantation, they were growing it for the hemp fibers to make rope. Because if you think back into that period of time, people were using sailing ships. And if you have a sailing ship, even today, if you have a ship, you want good rope. And hemp made very good rope. So it was a cash product to be sold. If you look inside this plant, it's not that different from any other I've talked about. It's got hundreds of different compounds. So no plant, just is made up of just one or two different chemicals. I think there's been 360 chemicals identified from the cannabis sativa plant, and of those, about 30 have some psychoactive properties. So they're fat soluble enough to get across the blood-brain barrier. And of those 30, the one that seems to produce the most potent changes in mood and has the most psychoactive effects would be this particular chemical, delta-9 tetrahydrocannabinol. You may see me abbreviating it as THC for the tetrahydrocannabinol. Sometimes you'll see the delta-9, sometimes you won't in the notes, but it's that chemical that I'm talking about. So there's 29 others that are psychoactive. That's why when someone is using the plant medicinally, it could be one of those other 29 that's actually giving them the benefit that they're looking for. This one gets people most intoxicated, but it may not be providing a particular medicinal effect that people want. And then there's all these other chemicals that are part of the plant but don't get across the blood-brain barrier. So if you look at this plant, has kind of an interesting history, because once we discovered that and were able to prove that the morphine and codeine in the opium poppy, we knew there had to be receptors in our brain that these plants were working through. Well, the same thing had to be true for the cannabis plant. How could we get intoxicated on a plant if the chemicals that we were absorbing from that plant weren't activating some system in our brain that we didn't already have? So that was the big race for a lot of research labs, is to be the first one to discover the receptors that THC interacted with. So there were, I think it was the lab in Johns Hopkins who won the race. I think one of the scientists there, he's already been named to get the Nobel Prize at some point or another. He's had a very big career in neuropharmacology. But in 1992, they did discover those receptors. And they did this by having radio-labeled THC. And it was actually injected into the rat, and then you could harvest the rat brain and look at where the THC was binding. Because you could put the tissue over top of radioactive film, and it would actually take a photograph. Wherever those radioactive molecules were, it would develop the film just right over that part of the brain. So first of all, it was very specific. So THC doesn't bind just everywhere in your brain. There are very selective brain structures where THC bound to. So that was kind of expected, because most certain receptors in your brain are not just spread evenly. Depending on what organ in the brain they're interacting with, that can give you a clue as to what the drug is going to do. So that was in 1992. Now, then the next thing was, if we have receptors for THC, what's the natural compound that binds to it? I mean, we wouldn't have receptors for a plant if we didn't already have something that activated that receptor naturally. So that's when people were looking for the endogenous neurotransmitter for these receptors. We had to have a natural cannabinoid in our brain that would activate these, just like we had to have a natural painkiller to activate our endorphin receptors. And so eventually, 1995, the first one was found, and it was named anandamide. So you do have to know that name, anandamide. It was the first cannabinoid that was discovered. We've discovered a lot of them since then. Once they discovered the technique for isolating these particular neurotransmitters, they've discovered a number of them. So they don't really necessarily name them now. They just call them as a group endocannabinoids. So endocannabinoids is sort of like an abbreviation of endogenous cannabinoids. So just like endorphins is an abbreviation of endogenous morphine, we now have the endogenous cannabinoids. So if you look at where the receptors for these natural endocannabinoids are, you can see certain patterns of where we think it's controlling function. So we've identified two different kinds of endocannabinoid receptors. The first one is the important one that's found in the brain called CB1, cannabinoid receptor 1, or cannabinoid 1. So I'm going to talk to you, there's three parts of the brain where these neurotransmitters are found. And when you see what part of the brain they're found in, it also helps explain some of the effects and side effects of getting high on marijuana. So there are lots of CB1 receptors in a part of the brain called the hippocampus. So you may want to try, these are all included in like a sentence format, but you could maybe make a little list for yourself. The 3 brain areas, we've got the hippocampus, we've got the amygdala, and you've got the hypothalamus. So those are the 3 brain areas. So what are those 3 parts of the brain? What are they responsible for? Okay, so I'm sorry I have a break in my page here, but what does the hippocampus do? Okay, well, if we look at the hippocampus is important in regulating short-term memory. So it doesn't have to do with you remembering that 2 + 2 = 4. That you remember forever. That is long-term memory. Short-term memory is what I've just said to you, and if you're trying to remember what I've just said to you, you're juggling the term hippocampus in your brain right now and short-term memory. You haven't deposited it as long-term memory anywhere. You're just using it at the moment. You've just heard me say it. You've just read it up here, and you're kind of juggling it around. That's short-term, sometimes called working memory. You're using it right now. So if I were to ask you, what function does the hippocampus have, some of you would be able to grab it out of your short-term working memory and say it has to do with memory, short-term memory. But 5 minutes from now, if you don't continue to try and use that information and move it into long-term, it's just going to disappear. So when people are intoxicated with marijuana, what's affected is their working short-term, what they just said, what they just talked about, what they just saw. So they tend to get a little distracted. So when I was in college, there was actually 1 phone on a dorm floor, and it was out in the hall. So if you wanted to call to get pizza, you actually, sometimes you would, and we didn't have, of course, working cell phones or anything like that, you'd look at the number in the phone book, and you'd walk to the phone saying 655-9923, 695-9923. That's working memory. If you're intoxicated with marijuana when you're doing that, you get about 2 steps, 695-226. Oh, man, you got to go back and look it up again. Okay, so that's the kind of forgetfulness that occurs with people who are intoxicated with marijuana. They also might do things where they're very intently talking about something, and then, what was I talking about again? They forget what the beginning of their sentence was. So that's people who would be, that's the kind of short-term memory I'm talking about. So it's not that your permanent memory, your long-term memory is damaged. It's just that when you're high, you're going to be acting a little bit hard to keep track of what's going on right now, what just happened. Okay, you tend to get a little distracted. So the second part of the brain that has a lot of receptors is called the amygdala. I may have mentioned this brain structure before. It often has to do with emotional responses to what's new in your environment. So animals and us, it's not something unique to animals, we tend to orient to things that are new that we haven't seen before. So if we see the same thing in a room over and over and over again, and then we walk into that room and suddenly there's a new picture on the wall, or there's a new couch or a new rug, we immediately orient, that is novel, that's new, that wasn't in this environment before. So under the influence of marijuana, people tend to be very sensitive to things looking new or different in their environment, even if it is something that they've seen before. They may look at a poster and then suddenly they're just amazed at this poster. Have you seen the colors in this poster? Oh my God, look at that. You can see where he's trying to, suddenly this poster is so novel and so interesting. So that's what some people enjoy about marijuana, is that it makes the normal look interesting again. It seems like things are now new, different, they have more meaning, and that's the amygdala that's playing a role there. Now, the problem with the amygdala is that it can also lead you into fear or anxiety, because sometimes if things are too new and different in your environment, and it can be exciting, it can be fun, but there's a part of the amygdala that can also get you to be afraid and say you don't like that something new and different could be bad, something you don't want. And so there are people who can often get paranoid when they're under the influence of marijuana. And that's a role probably the amygdala plays there. It's like things either look different and new and exciting, and that's good, or they look different, new, and then bad things could happen because of that, and you get paranoid. And then finally, the third part of the brain, the hypothalamus. has to do with triggering your interest in food. So why do you eat? Well, it makes sense that you would eat because you're hungry, you need calories. So how does your brain tell you it's time to eat? I mean, we all think, well, but remember, there's no little man in our head that says, time to eat. There's parts of your brain that monitor blood glucose and all kinds of things. And when it's time for you to eat, it will cause you to be more attentive to food signals. So even with you being unconscious of it, suddenly you smell pizza, you smell fresh bread, and you orient to that. Wow, that smells so good. Now, if you were not hungry, that same response could be out there, but you'd smell it, but it's no biggie. I'm not hungry, I don't want to eat. So this is how this part of the brain sort of steers you towards eating. It controls eating behavior. And guess what? It has a lot of endocannabinoid receptors. So this explains an old behavior that everyone talked about called the munchies. They'd be talking about getting high on marijuana, and then about an hour later, oh, wow, ice cream. Oh, milkshake, this is from heaven. This Mounds bar is just the best. You know, everything just tastes so good, you just like to eat. The munchies is basically what they said. And we couldn't really explain the munchies. It's not very well replicated in the lab because rats don't express munchies the way humans do. But we now understand it completely because you're activating these cells in the hippocampus so that the sense of food, the smell of it, the sight of it, the taste of it is much more interesting. And so you're drawn to it, to eat. So it kind of makes sense now when people are intoxicated with marijuana, can make things look new and novel, can make you, you're focusing so much on what's happening in the now, but you can also kind of lose what you just said, the beginning of your sentence. You're kind of orienting to things a little different because of the short-term memory changes. And at some point, food may seem very tantalizing to you. So, so those are the kind of things that people get intoxicated will experience. Now, there are some cannabinoid receptors that are found in the immune system. We're not going to talk about those. There are some that are related more to also out in the body that might play a role with relieving certain kinds of pain that people feel. But there are medical uses even for some of these, particularly this increased sensory appeal of food to help people who have cancer and they're undergoing chemotherapy. Sometimes they absolutely cannot eat. In fact, smelling food, they get nauseous. So there are some medical uses for helping people eat when they're undergoing procedures that really make them kind of nauseous. And if you can't eat, you're not going to recover from your illness. So those are pretty big advances in terms of what exactly marijuana is doing. If we look in terms of is there more than one kind of marijuana out there, it's a little bit of a controversy, but some people think there's actually 3 species. Some say it's still just one, and then there's just variants there. But the most well-known is cannabis sativa. And then there is a different variety, and I think it may be actually a new species at this point, called cannabis indica. So one of the things that's happened is that when people went from, when it was illegal in the United States to be growing marijuana, and if you wanted to grow marijuana inside, there's an advantage to having small plants. I mean, if left on its own, particularly the male cannabis sativa plant, it'll get 10 or 12 feet high. And a 10 or 12 feet high plant is pretty hard to conceal. Even people that are growing it out in the forest, they don't want it to get tall enough where you could see it, with a helicopter going by or things like that. So there was sort of a push to have smaller plants that you could grow inside. And that's one of the characteristics of cannabis indica. It is a shorter plant, it's a bushier plant, and even the leaves are not as long and center, they're kind of also shorter leaves. And it's grown more for the intoxicating effects. You wouldn't grow cannabis indica for hemp fibers, okay? You would definitely want to go with a tall plant to get the fibers. But here, people are concentrating on the intoxicating parts of the plant. So they've been interbreeding, just like you can do with tomato plants and corn plants and tobacco plants. Humans can experiment with plants by interbreeding different varieties to get what they want. And so one of the versions of cannabis indica is called skunkweed. And it's because it has a lot of THC. It has a lot of, the female plant has a lot of resin, sticky resin, where the THC is found. But the resin also has a very strong scent. So if I had some skunk weed in my pocket, even if I had it double wrapped with a baggie and tinfoil, probably by about now, people in the front row there would starting to smell something that would be like a skunky odor. So it's kind of hard to cover up the odor of skunk weed. But it does have a very, very high level of THC. So I know they grow it here in the hill country. Right about now, it's being dried. Oh, no. No, it's pretty much starting its growing season. Usually, if you get a gift, I usually get a gift at some point during Christmas, of a little tinfoil wrapped thing of skunk weed. I'm not sure who the person is, but they always get it to me somehow. And it grows in the summer, it's dried in the fall, and it's harvested. Well, it's harvested in the fall and dried. So this is, again, something you wouldn't be, if you were to light up a joint with skunk weed, the neighbors would know. That's one problem with it. Okay, so those are the two, sativa and indica. There is one other one that grows only in Siberia, doesn't have a lot of THC, Cannabis ruderalis. You don't have to worry about that one. Okay, so if humans are experimenting on how to get the most intoxicant out of the plant that they have, I will say that there is another thing that people do. You may have noticed when I sometimes would get pot when I was in college, it had seeds. Okay. Now, if you buy marijuana today, you probably will notice that if it has any quality at all to it, it does not have seeds. And that's because people have adopted this growing technique to produce marijuana that is sensimilla without seeds. Okay. So how do you get pot that doesn't have any seeds? Well, what you do is when the plants are very small, you pluck out all the male plants. And you can tell the difference when you study the plants a little bit. So you get rid of all the male plants. So the female plants are growing, they get big enough, they're mature enough, they start to have a little flower, and they want to be fertilized by a male plant. And so they're waiting for the wind or a bee or something to bring something to them to fertilize their flowers, and nothing happens. So what happens is the female plant just keeps putting more resin into her bud, getting it stickier and stickier and stickier, thinking, well, I'll get somebody now. Never happens because there's no male plants. So when you harvest those female plants, they don't have any seeds because they've never been fertilized, and the flowering bud is all covered with sticky resin. And THC is located in the resin. So there you are. You have now harvested a crop that has a higher percentage of resin per plant material, which means more THC per plant material, and no seeds, because if it's properly done, none of those female plants have been fertilized. So that's why you may see now that it's unusual to get marijuana with seeds in it, and if you do, you may notice that it's not a very good quality. They did not use this growing technique. So that's just one of the reasons there has been some increases in the percent THC in the plants. This number is all over the place. This includes some hashish and other preparations. I'm not going to ask you about the specific percent content, so don't worry about that. But just know that there are different potencies of marijuana out there. India is another country where a lot of marijuana is grown for intoxicating effects. I think in your textbook there's some pictures of some monks who are making a preparation of some tea. They're heating it up and trying to extract the THC, and they'll drink it as part of their ceremonies. So they're aware of different levels of potency. These are names that sometimes have been borrowed in the United States. In India, bhang is usually that low quality marijuana. It may have some seeds in it, some stems in it. Ganja then would be the next level up, where you're just concentrating on those flowering tops. And then hashish is where it's a process where you kind of grind it up, get rid of as much plant material as you can. So what's left is mainly a brick of resin. So hashish is usually in little bricks, could be like a thick piece of chewing gum, and that would be mainly THC, so you don't need a lot of it, and most of the plant material has been removed. And then actually there's another level called hash oil, where you go ahead and refine it even more so that all that's left is kind of a black oily resin, and that contains just about all THC. I mean, that's where it's really concentrated. There isn't any plant material there. So what people are doing is they're trying to concentrate what they have. All right. Now, I always have a lot of people ask me, about drug testing and marijuana. And so I did want to just mention a couple things. We'll start out with urinalysis. And I do want you to know that I would say 99% of the time that what they're using nowadays for looking for the metabolites of a drug that you have consumed. So I had mentioned to you that no drug stays forever. Every time your blood goes through the liver, some of the drug molecule is broken down into fragments called metabolites. And those metabolites have a specific shape based on their original molecule that they are fragments of. So most of the time, the urinalysis kits are going to use color-enhanced immunoassays. So an antibody has been made to a fragment of a specific drug. And so the thing about antibodies is they're very specific. So they will only interact with one type of fragment. So usually the company has to pick 3 drugs. They can't test you for everything because it would be so expensive to have an antibody kit that had antibodies to everything. So it's easy to pick marijuana. So maybe Valero or SeaWorld, they say, well, okay, we're going to do a urinalysis. We'll pick marijuana because these are young people. Then they'll pick usually one painkiller. They may go with like heroin because that's a known illegal painkiller. Those antibodies will not necessarily react with OxyContin or Vicodin, but it's always good to tell them if you have a prescription for a drug. And then they have to pick a stimulant. So they may pick cocaine if that's the hot thing that's out there, or they may pick methamphetamine if that's the hot thing that's out there. So they're going to pick 3 antibodies, and that's what's in the kit. Then they take your urine sample, they split it in 3 parts, and then they'll test those. Well, they've been trying to get these kits to be easier and easier and faster. So now they've got ones where they just dip a stick into the solution and look for color bands. The color reaction is that when the antibody binds to its target, a color molecule, a color chain reaction occurs, and it might turn blue. So now they've got it down to, I know they can actually do a saliva test. They take a swab of your saliva, and they're looking for reactions there. Well, I think each of those, as they try and get quicker and faster, because it's expensive, I mean, they have to pay for each of those things that they're doing, is they're actually not as sensitive. I mean, something like marijuana on a saliva test is really dumb, because I don't think there would be any marijuana metabolites in your saliva. They're looking usually there for things like alcohol or maybe one other drug they can look for in the saliva. But they are not, remember the tests have to be set to a certain level of sensitivity. You can make these kits so sensitive, that means there's so many antibodies in there that they might start cross-reacting and everybody turns out to be positive. Everybody turns blue, which would not be a very good test. So they're going to back off on the sensitivity till it's down around, for marijuana, about 50 nanograms of metabolites per 1 ml of urine. So that means that you could have below that and be fine. And at this point, if you are not a regular marijuana smoker and you smoke a joint, you will be fine in 24 to 72 hours, okay? The people who have to worry about urinalysis tests are the ones that are chronic users of a drug, which means that they use the drug every day as part of their life. So they're the people that wake up, hit the bong, take a pipe with them or a one-hitter. They're smoking pot all day. When they get home, they're smoking more pot. In fact, if they're not getting high, they don't feel right, you know. And when they stop using, they miss marijuana. Now, for people who are chronic users, now you're going to take a while to get those metabolites out of your blood. So it might take a month before you get below this test level. And for you to get to 0, it might take, another month or 3 or 4 months. So they won't test down to 0. If you're an Olympic athlete, they'll take the test down to, I think, 12 nanograms per ml, which is very sensitive. But I do want people to recognize that this whole idea that it's in your body for months has really gotten blown out of proportion. Because I have had people who smoked a joint during spring break, and they're still calling me going, will I pass? I'm getting a job interview at Valero. And I'm saying, you are fine. If you're telling me the truth and you just had a joint and you're not a regular smoker, you'll be fine. But what happens is people are setting up this billion-dollar business now where these companies sell this crap that people guzzle it and get sick and all that kind of stuff. And then they pass their test. And they're like, oh, you got to use this stuff. Or make sure you pass your test. Well, guess what? You would have passed anyway. So all you're doing is putting money in the pockets of these people now that are making billions. The price of this stuff has been jacked up now. And think about it. What could you drink that would actually get stuff out of your blood? It's not like there's anything you can put in your intestines that's going to suck something out of your blood. It doesn't exist. So what's happened is because people drink this crap and pass, then all their friends are going to drink it and pass, not realizing that they would have passed otherwise. So my big problem with urinalysis is, first of all, well, for one thing, we don't have a good test for what the biggest problem is, and that's alcohol. Because people hungover at work actually are the most dangerous. But they're not going to have alcohol in their blood or urine. The other thing is, if someone is doing something on their own time, I'm not sure, again, how that is going to be something that you would say, well, this person is not fit to do their job. I also feel that these companies go out to the lowest bidder. I've seen some of the techs who do these tests. I wouldn't want them to mow my grass. They don't look very competent. Okay, I'm going to take these over here now. They're not paying these people that much. Who knows if they're even doing the test correctly? They don't keep it at the right pH. They don't keep it at the right temperature. God knows they don't keep them labeled correctly. I mean, I had to do a lot of urinalysis. I did some, what's it called? It was for the Air Force, for getting pilots so they could wake up and go to sleep at the right times. But it was something that they wanted, you had to get clearance. Okay, it was classified research at Brooks. So, I'm doing urinalysis all the time, and I see these people back there that are doing it, and I'm thinking, what if they get my stuff mislabeled, which is very common. People have known to be paid techs to shuffle labels around. That's why you get the lowest bid companies. They're not the best. One time I failed because my urine was too dilute. So I'm down at this crummy little clinic downtown. I'm waiting for 4 hours for urinalysis. I'm going, oh my God, finally I can urinate. And of course it's very dilute, because I had very dilute urine at that point. And so then I got to go back, because they said, well, maybe you drank a lot of water to dilute your urine, so we wouldn't detect. I said, oh, that's fine, okay. Well, the next time you go back, you're in the special group. So I'm sitting there with all the real guy next to me. He's all tattooed up with alcohol on his breath going, what are they going to test you for? I said, well, hopefully they're not going to test you for booze. I mean, he was already drunk. But there has to be witness. So then you have this little Nazi that has to watch you urinate into your cup. And I'm thinking, just let it be over. And you actually get paranoid. I'm thinking, I know there's nothing for me to be afraid of, but you get kind of paranoid that they're going to find something. And I've also heard that Susan Reed in the DA's office throws away samples because they have so many, millions of them. So if they did make a mistake and have a false positive, which means they found something that you didn't have in your urine through some mistake, you couldn't even go back and have it retested. So that's led to a lot of problems. So, I think that urinalysis and testing has a place. And the people who have a problem with drug testing are usually people who have a problem with addiction or abuse, because they can't control their intake. Now, the hair follicle or the hair thing, the whole idea of that is, and I have my doubts about how sensitive this could be also, is that every hair follicle has a little capillary that nourishes it. So and hair is continuously growing, even the hair, fine hair. But there's a certain rate that hair grows. And while it's growing, if there's a chemical or a compound in the blood that's nourishing that hair follicle, it could be embedded into the hair. So that's how they've been able to dig up some people, and they found arsenic embedded into their hair follicle, and they knew that these people had died because of arsenic poisoning. Well, theoretically, if there's a lot of drug metabolites circling around that hair follicle while it's growing, some of those could get embedded into the hair shaft, just by accident as it's growing. So then, what they're saying is, okay, they can pluck a hair, and I actually talked to somebody. She was in my class. She was actually the person that had to do the plucking. She said, oh, it's terrible, So, you think about the amount of material that's there. It's very small. You're going to dissolve that hair. so that all that's left is a solution of the parts of the hair, it's chitin and protein, and then some metabolites they're looking for. I would think you'd have to have a pretty big dose of that metabolite in the hair to really get to know, be able to say, okay, there was a definite positive here. I don't know how many positives they get, but I do think it frightens a lot of people that they smoked pot 6 months ago and there's going to be a little thing in their hair that's going to give it up. I can't imagine if they have that sensitive a test to do it. So again, I think what you're getting is people who are heavy chronic users. And if that's the case, then they should just say that up front, instead of scaring people who may have used a drug once and now they're afraid they're going to get caught. So I think that most of the time they're going to miss things because they're looking for something that's a very small amount. And most of the people aren't even at that level. So that's my saying on that. So just be careful. Where I would drug test is if somebody was in rehab. If they're in my clinic and I'm taking all the time to try and treat them to get them over their addiction, and they're still using, they got to get out of there. So I would definitely drug test every day anybody who was in a rehab clinic, because if they didn't really stop, if they're just lying, you don't want them in your clinic. They're just taking up space. So that I could see. But to drug test everybody, not a very wise thing to do. All right, just want to make one quick mention about using these effects of marijuana for medical reasons. There is a pill. The trade name is Marinol. That pill has synthetic THC or another. I know there's another cannabinoid they put in there, and it helps people who have no appetite to eat. So usually they're going to give it to people who are on chemotherapy or who have very serious wasting diseases that they are they're going to help with helping them to eat so that they won't get too thin. Why did you guys even bother coming? There's about 5 minutes left to the lecture. Must have had a bad bus ride or... Okay. Wow. All right, so Marinol is still out there, and some patients will take it. But a lot of patients say that they don't like Marinol because they can't control the dose. And one thing about using the plant material, if you know what works for you, you can decide whether you have enough or not enough. And with the pill, you're not always sure whether you're getting, if it's too much or it's not, because you will have the side effects of intoxication while you take the drug. Okay, well, I just wanted to remind people that I discovered before I left the house this morning that the quizzes didn't open up, so I apologize for that. I didn't have time to do anything about it. I'm going to go back to my office, sit down and find out why the quizzes didn't open up, get them open, so they'll be open until Sunday. I didn't have a chance to add your points in from Friday, and I didn't have a chance to, it was a bad weekend, and I didn't have a chance to do any extra credit from the list that's now available at ASAP. So all of that I'm going to be working on this afternoon. So if you have a question about that, that's what the take is on that. I just didn't feel very good and got way behind. So I'll go ahead and get set up here, see if we can ask a question. Okay, I'm actually going to have to write one out for you real quick. I'm just writing out your question here. This is going to be a handwritten question. I'll get it up here in just a second.